Assalamu alaikum and welcome back to my YouTube channel Studyholic. I'll be discussing about lysosomes in today's lecture. As I've discussed about Golgi complex in the previous lecture, you can watch the video if you want to get the concept. And let's begin with our today's lecture, that is lysosomes. So for lysosomes, for understanding what actually lysosomes is, we would have to break the word lysosomes into lyso and zomes. The word lyso means splitting and zomes means body. Sorry, I can't write this split hole. So its function is explained itself in its word, that is splitting the body. I'll be discussing about its further detail in the lecture, so stay tuned till the end. Now let's discuss about lysosomes. These are single membranous spherical bodies, and the lysosomes basically contain lysosomal enzymes, and the amount of lysosomal enzyme is about 40. They contain about 40 different digestive enzymes because they break the body, and in digestion breakage takes place, so it contains 40 different digestive enzymes, and these 40 different digestive enzymes are capable of breaking down every major macromolecules of the cell. So, if we talk about lysosomes, lysosomes are basically present in animal cells, whereas in fungi and plant cells, certain vacuoles carry out the function which is carried out by the lysosomes in animal cell. So, if we talk about the contents of lysosomes apart from enzymes, their contents are acidic in nature. Now let's move on to the formation of lysosomes, where lysosomes are actually formed. You can watch my previous lecture about Golgi complex. There I have also explained regarding lysosomes, so you will better understand in this lecture. Here is a diagram which I have attached from the book. Now let's discuss about the formation of lysosomes and lysosomal enzymes via this diagram. Here it is rough endoplasmic reticulum. The lysosomal enzymes are formed in the rough endoplasmic reticulum. Lysosomal enzymes are also known as hydrolytic enzymes because they are gonna break down the molecules further. So I have written over here that lysosomal enzymes are formed in the rough endoplasmic reticulum, and then they are transported via vesicle through smooth endoplasmic reticulum into the Golgi complex. You can watch in the previous lecture about the Golgi complex how the lysosomal enzymes are processed into it, and then the lysosomes are excreted in the form of the vesicle through the trans phase of Golgi complex. The vesicle, basically the enzymes are processed into the Golgi complex and they are excreted in the form of the vesicle, and that vesicle in a result is known as lysosomes. And the newly formed lysosomes, which are excreted or transported through the Golgi complex, are known as primary lysosomes. That was the formation of the lysosome, how the enzymes were released from the rough endoplasmic reticulum, processed into the Golgi complex, and the lysosomes were formed via the Golgi complex, and that lysosomes which were released through the Golgi complex are known as primary lysosomes. Now let's discuss about the types of lysosomes. There are basically two types. The one, primary lysosomes, second, secondary lysosomes. Primary lysosomes, as I've discussed earlier clearly, that the newly formed lysosomes are basically known as primary lysosomes. I have to come back to explain to you regarding the secondary lysosomes. Till here, the lysosome is known as primary lysosome. The lysosome has to break down the food particles. So the food particles are entering into the cell as a food vacuole, and they are named according to their mode of entrance. There are basically three modes of entrance, that is endocytosis, phagocytosis, or autophagocytosis. And if a food vacuole is entered into the cell via endocytosis, we will regard it as endosomes. If it enters via phagocytosis, we will regard it as phagosomes. And if autophagocytosis, it will be regarded as autophagosomes. I have mentioned over here, if you can understand my writing, this is endosomes, phagosomes, and autophagosomes. So, how come the secondary lysosome come into being? In order to perform the function, the lysosome has to fuse with the food vacuole. And when the primary lysosome, the newly formed lysosome, fuses with the food vacuole, the secondary lysosome came into being. And when the secondary lysosome is formed, it results into the further breakdown of biomolecules, and the smaller particles that led after the breakdown are known as amino acids, monosaccharides, nucleotides, and fatty acids, which are again recycled into the cell. As you can see, the secondary lysosome is formed, and this will lead into the digestion of the food. The macromolecules are broken down into the micromolecules. Now that was the formation and particular the mode of action of lysosome. Let's discuss about the functions of lysosomes. There are basically three functions that are intracellular digestion, autophagy, and autolysis. Let's discuss them in detail. Okay, so the first one, that is intracellular digestion, is the same that we have discussed earlier, that how food vacuole is come into the entered into the cell, the lysosome fuses with it to form the secondary lysosome, and as a result it leads to the digestion. So the digested products, that are amino acids, monosaccharides, fatty acids, are basically recycled in the cell, and the waste products are remain into a vesicle, and that is called as contractile vacuole. Okay, I have drawn a flow chart over here. First of all, here is primary lysosome. That primary lysosome, when fuses with the food vacuole, it results into the secondary lysosome, and then the digestion begins. After that, the digested products are recycled into the cell, whereas the waste products are accumulated into a vesicle, and that is known as contractile vacuole. So I hope you got the concept regarding intracellular digestion. Now let's move on to the autophagy. If now also you have some confusion, feel free to ask me in the comment section, or you can also email me. Now let's talk about the autophagy. Here is autophagy mentioned, and here is also a small diagram illustrating about autophagy. Let's talk what is actually autophagy. The process by which unwanted structures within a cell are engulfed and digested within the lysosome is called as autophagy. Autophagy process is basically known as self-eating process, in which the lysosome begins to eat the cell's own contents. And when does it happens? First of all, let me know that the lysosomes which eat its own cell products are known as autophagosomes. This process, the autophagy process, basically takes place in starvation period. It takes place either in the starvation period or either in order to control the number of specific organelles. In starvation period, it is taking place to obtain the energy. Let's talk about an example so that you can better understand about it. If a person performs heavy exercises, uh, the mitochondria accumulates, mitochondria count begins to increase in its cells, in his muscle cells basically. And when he leaves the exercise, the number of the mitochondria are again brought about to the normal by the autophagy process, because the lysosomes eat up the extra mitochondria. So here also they're explaining about the mitochondria, how the lysosome engulfs the mitochondria and it is designated as autophagosome, and then the digestion begins over here. Now let's talk about our last topic, that is autolysis. Autolysis process basically takes place during developmental phase. It particularly takes place when a particular cell is required to be eliminated or disintegrated, and then the process by which that cell is eliminated is known as autolysis. And this type of cell that basically is known as autolysis. So how does this takes place? Let's draw a cell. Here is a lysosome. These are the contents. Let's suppose here is enzyme, right? Let's imagine. And here are all the organelles, the endoplasmic reticulum, leave it, all the organelles. So when the autolysis has to take place, what happens? The lysosome basically bursts, it bursts, and as a result, so what happens when the autolysis has to take place? The lysosome basically bursts, and when it bursts, the contents leak out of the lysosome as a result, and this results in the digestion of cells' own contents and ultimately cell death. As a result, the cell is disintegrated into fragments, and that fragments are phagocytosed by other cells. And due to this function of the lysosome, the lysosomes are also known as suicidal bags. That was all about the functions of lysosome. Let's move on to our last topic, that is lysosomal storage diseases, and one of that disease is Tay-Sachs disease. Lysosomal storage diseases are basically hereditary and congenital, and therefore they run in the particular families and exist by birth in an individual. Most of these diseases are fatal in early childhood. About more than 20 such diseases have been discovered so far, but I'm going to explain only one example to clear the concept, that is Tay-Sachs disease. How the Tay-Sachs disease reacts. When the lipid digesting enzyme is missing or inactive, so this the lipid digestive enzyme, the absence of the lipid digesting enzyme results in the Tay-Sachs disease. Why I have drawn the brain over here? Because due to the absence of lipid digesting enzymes, the lipid starts accumulating. There's all over lipids in the brain, and when there is the accumulation of lipid in brain, the brain becomes impaired, and this results in the Tay-Sachs disease. I'm not gonna go into further details into the signs and symptoms of the Tay-Sachs disease because it's not your requirement, not requirement at this level. So you can write down all the notes if you want, and you can also ask me. I'll help you out in email regarding any concepts. That was all about today's lecture. Let's move on to our favorite session, that is brainstorming. Lysosomes are also called as vesicles, food vacuoles, contractile vacuoles, suicidal bags. I know the BCQ is not very difficult, you would be knowing. The answer is suicidal bags, as we have discussed earlier in autolysis. Let's move on to the next one. Impairment of the brain due to the lipid accumulation leads to Klinefelter syndrome, Turner syndrome, Tay-Sachs disease, Down syndrome. Yup, the answer is Tay-Sachs disease. So I hope you like the video, and don't forget to like the video and share it with your friends, and please subscribe to my channel. Thank you. Stay tuned.